Good afternoon, everyone. Hope you can hear me. Today we have an extra seminar, which is not in the original schedule, but we just happen to have a guest in-house for a couple of weeks now. It's Professor Ximena Abrevaya. Sorry, I mangled your name, most likely. She's from Argentina, and she's a senior research scientist and astrobiologist at the Instituto de Astronomy y Física del Espacio, okay, sorry, in Buenos Aires. And she's also the founding director of the Argentinian Research Unit in Astrobiology. And she did her master's and PhD in biological science at the University of Buenos Aires. And her PhD thesis was the first thesis in astrobiology in Argentina. And she is quite often here in Austria, in Graz, as a guest scientist at the IGAM at the University of Graz. And we've met already a couple of years ago, and we had always discussion if we could do some common experiments. And the last couple of weeks, we made this happen, and we did, I think, 4 weeks in total of astrobiology experiments here in-house in our astrolab. And she will represent part of her work she does in general and also reflect on what we did here in Graz. So, what's your place?
Okay. Thank you for the invitation for the seminar. Yes, I decided to choose this topic to talk a little bit about the interdiscipline because I was starting to work in astrobiology in 2007. Before I was working in a different field. And what was motivating me to work in astrobiology was to think in the idea of combining different areas, in particular to improve astrophysical studies by the addition of some biological experiments. And in 2016, with other colleagues in Argentina and following all these interdisciplinary collaborations we were having, and I was having with different persons in Argentina, we decided to create this Argentinian Research Unit in Astrobiology, which is involving different people from different institutions in Argentina and different backgrounds. In green color, you see the people with biological background. In light blue, with physics or astronomical background, and in orange, people with background in geology, and in yellow, people with background in chemistry. And also we are having external collaborators and Austrian researchers are making an important part of it with Professor Hanselmeier, Dr. Leitzinger, and Dr. ÖAW. We are having a very long-term collaboration since 2012. They are making an important part of all the studies we are doing. Very recently, as Gunther mentioned, sorry if I wrote your name in the wrong way, using the umlaut and the H is wrong, sorry. We are starting a collaboration this year, as he was mentioning. And yes, we are having also collaborators from other countries. The main research topics we are currently developing, they are related to two different kind of environments. We are on one side studying highly irradiated environments in planetary surfaces at levels which are higher than the terrestrial levels. This is what I mean with highly irradiated. And we are studying the influence of radiation on life. In particular, the influence of astrophysical events on life. And with this, we are having two programs or projects. One is the BioSun, and the other one is the EXO-UV. I'm going to mention something about this in this course. And the other area is covering the hypersaline environments, where we are studying the influence of salt minerals on the protection and preservation of life or molecules. This is connected with another project we are having. And there is also a connection between both topics, because some of these hypersaline environments are also highly irradiated, and the microorganisms we find in the surface, particularly in surface hypersaline environments, are as well having mechanisms to cope with highly irradiated environments. So I will mention something about this in the talk. But in general, one could say that both topics are fitting in the topic of habitability, and yes, in the analysis, if the fact if an environment can or cannot sustain life in its habitat. Well, while we are studying radiation, one important point is that the stellar radiation, the UV range is particularly interesting because it can reach the surface of the planet, and you could have effects on different levels. You can have effects at the level of the prebiotic chemistry. There are some studies indicating that UV could influence in prebiotic reactions. It could have influences in prebiotic, having the effects which are related to microorganisms or organisms. And then we are having effects at the environment, typically at the level of the atmosphere. And during this talk, I'm going to mention our studies which are mostly connected with effects of the level of organisms. And for that, there are, well, biologists, we used to divide UVC in these ranges, and UVC and UVB are the most relevant because they are the most harmful for life. But not only planetary surfaces are interesting to study in the context of radiation. We have also processes as lithopanspermia, particularly the interplanetary travel step, which involves the exposure of the meteorite to very high levels of radiation in the interplanetary medium, and also for usually for extended periods of time, sometimes of million years. And now the question is, well, how we study this biological impact of radiation from astrophysical sources. And there are several possibilities. One is to make theoretical studies, but the other possibility is to make experimental studies. And I divided this in two different kinds of experiments, which are possible and existing. One of these experiments are space experiments in real environmental conditions. Typically, these are experiments performed at the low Earth orbit. For instance, experiments running in the International Space Station. And then we are having the other possibility, which are the laboratory simulation experiments. We simulate environmental conditions which are similar to the one in other planets. And there it's possible to use facilities or instruments for these simulations. And in our case, we are particularly focused in these kind of experimental approaches. Well, one could think in advantages and disadvantages of every of these approaches. This is something probably we can discuss at the end of the talk. But in general, under my point of view, what I see in astrobiology is that we have a proportion of studies which are astrophysical studies, which are, contains predictions about life survival. These are usually astrophysical studies. And then in these astrophysical studies, they make some kind of predictions about what could happen with the organisms related to these astrophysical events. On the other side, we have also biological studies, which are predicting the survival of microorganisms exposed to radiation. But sometimes these kind of studies are having no or weak correlation with the astrophysical events or information. So our approach is a combination of both. And so we are using theoretical or observational astrophysics. And then we apply these to these simulation experiments in laboratory conditions where we could control parameters, for instance, as temperature, atmospheric composition or vacuum, and radiation. And as I was mentioning at the beginning, we have two different projects related with radiation. One is the BioSun project. This project is connected with the origin of life and the influence of radiation in the origin of life or the first life forms. And here we are studying particularly solar stellar analogs and the EXO-UV program, which is connected with exoplanets. And the goal of this projects or program is to combine different fields to expand and improve previous studies by the incorporation of these laboratory simulations. And well, we have these high energy emissions from astrophysical events, and we are focused particularly now in flares and super flares. And why is it relevant to study this? Well, this is because there are few studies which determine the biological impact of these events in planetary bodies. Because also previous studies estimated the biological impact using theoretical approaches, and they use also information from the literature about the biological impact, which from experiments which were made at low fluencies or low fluence rates. And this is a problem, as I will explain later. And why to focus in the UV range? Well, as I mentioned before, because it can reach the surface, the planetary surface. And also, I was hearing these arguments. Well, the biological effects of UV are very well known already. They are studied since centuries. But yes, in this case, it can be different because most of the studies you see in the literature are related to these experiments, which in comparison with an astrophysical event are having low fluence rates or low fluencies. And this makes a difference, as I will show. And also, some other persons say that the UV is not really relevant because you can have life anyway, and that the life can exist below the surface and protected, of course. This is true. But we are looking for, we are looking for the surface habitability, and this can be particularly relevant now because we are looking for exoplanet signatures, biosignatures, and we expect that these life forms probably are on the surface of the planet to bring these biosignatures we are going to observe. And well, there are very complex mechanisms related to the damage of UV. And as I was mentioning before, UVB and UVC are the most damaging. And particularly UVC is very damaging because the DNA molecule is absorbing directly radiation from this wavelength. And this is something you can see even in this plot. You can see the absorption of proteins and DNA, and they are at very short wavelengths. So that's why they are very damaging. Yes, the question now is, okay, what kind of stars we are going to consider in our studies? Well, usually in astrobiology, we are focusing considering exoplanets in F, G, K, and M stars. We could also consider A stars, but these are in the very limit, and this is because we take as a parameter the time which took life to develop on the Earth. So the minimum time would be 1 giga-year, but this is quite a few time. So then we consider these other stars. Well, this is the first example I wanted to show you. This is a study we published in 2020 related to this EXO-UV program, where we studied the impact of stellar flares on life using experiments. And then this year, and this work which was submitted to monthly notices, we are also studying the second case of this program. Is a study based on TRAPPIST-1 and particularly planets E, F, and G, because these planets are considered that they are in the habitable zone. So, well, first, before showing what we did, I wanted to tell you a little bit which were the previous results from other authors about these planets and Proxima B and TRAPPIST-1, E, F, and G. Well, this, as I was mentioning, these studies used theoretical approaches, and well, for instance, in this paper, Proxima B, they were taking a super flare case, and normally they use this, which is called lethal dose 90. I will explain now what is this. And in the other study, TRAPPIST-1, they were using this F10, this is fluence 10. This is also this the same, just is a different way to call it. The lethal dose 90 is the dose or fluence of radiation you need to kill 90% of the population of microorganisms. And the F10 is saying the same. The only difference is that this is considered 10% of survival. So it's exactly the same, it's a different way to call it. And it's very important, this fact that they use this parameter because of something I will explain later. This is what is called a survival curve. You measure here the survival fraction after exposing the microorganisms to radiation as a function of the fluence or dose. In case of UV, we are talking more about fluence. And this parameter is taken from the slope of the survival curve. And well, for the Proxima B case, previous studies predicted that there would be no survival on the surface of Proxima B, or at least it will be very difficult. You will need very complex adaptations of life. Given that the maximum lethal dose, I mean the lethal dose 90 is 553 joules per square meters. This is meaning that if you have much higher fluences than this, like these ones would correspond to a super flare, you will have survival. And the same applies to the TRAPPIST case, where they use this value for the Enococcus and also for E. coli, which is the Enococcus radiodurans is a very radiotolerant microorganism and is used very frequently in astrobiology as a model. And here E. coli is a bacteria also, but this is susceptible to radiation, is sensitive to radiation, so it's used as well as a model of microorganism which wouldn't resist too much to radiation. And in this case, they were considering different scenarios, like if you will have oxygen. So what they predicted is that E. coli will survive in any of the planets, E, F, and G. And if you consider the Inococcus radiodurans, no survival on planet E, but survival on F and G. If you have an ozone, you will have survival, but this is because the ozone layer is shielding most of the harmful radiation wavelengths. And then if you have an ocean, they predicted for E. coli that you will have only survival if the microorganism is below the surface. And for the Inococcus radiodurans, they predicted that would be survival. And also some other studies were talking about the need of having UV protective atmospheres based in the ozone to have survival in these planets. And now I will show you what we did in our studies. Well, very briefly, just to mention that we studied the UV in quiescence for both cases, and I was mentioning before, and the quiescent levels are negligible for microorganisms if we compare with the radiation levels of the microorganisms are receiving on Earth, or with the experiments which were done at the low Earth orbit, where the microorganisms were exposed to UVC. And we also made another study which involves the fact of considering a different kind of atmosphere, which is not also not what will happen if we have CO2 or nitrogen atmospheres. And these kind of compositions would be, in principle, more stable in planets orbiting these M dwarfs, not like oxygen. So we decided to study this case, and we saw with different pressures and CO2 compositions that there will be shielding as well to UVC and UVB as a function of CO2 content and atmospheric pressure.
Could you explain the colors? So what are the pressures?
Yeah, sure.
I can't read it.
This is irradiance as a function of the wavelength, and the colors are different pressures. I don't know, well, the black one is no atmosphere, and then you have from 1,000, well, here I see even less, is I think 1,000 millibar to 5,000 millibar. So it's increasing, the pressure is increasing in this direction.
So five bars is the last curve, yeah?
Yeah. So you see, if you increase the pressure, you have more shielding, particularly for the most harmful wavelengths, which are UVC and UVB. And also here is increase in the CO2 composition. So you have here CO2, pure CO2, and here 10% CO2, 90% nitrogen. So as more CO2 you have, more is shielding, and as higher the pressure, more shielding. Yeah. So now I'm going to the part of the flares and super flares and the experiments. I am just making a summary because it would be very long to explain both the studies, but I'm trying to make a summary of everything, of the most important parts. Anyway, we can talk after the presentation, and you can ask me more questions. So what we did in the lab is to reproduce or try to reproduce the UVC fluxes from the super flares and flares. In the case of the Proxima B, we took a typical flare as a case and a typical super flare as a case. And well, we were using different fluencies up to 40 kilojoules per square meter. Yeah, and we were working with different microorganisms. In principle, for the study, we included Pseudomonas aeruginosa. This is a microorganism which in principle is not tolerant to radiation, or we thought is not tolerant to radiation as the Inococcus. And Halopherax volcani, which is halophilic archaea, microorganism living in hypersaline environments, which is tolerant to UV. And for the other study, we were also considering the UV fluxes from the flares, but in this case, we consider the complete range from 200 and 400. And we converted this to equivalent fluencies of germicidal radiation. This is the radiation we use for the experiments, which is this wavelength, because it's very damaging. And we consider here a super flare case with different fluencies up to higher levels than in this study. And here we consider E. coli and Deinococcus radiolumens. And later we also performed another study with the same values, also experiments with Deinococcus radiolumens. And the irradiation conditions were we irradiated in a thin layer of water, like the cells were in water, suspended in water, and there was mixing to prevent that there is shielding between cells. And also an important part is we consider the unattenuated fluxes, so that we didn't consider that there is no shielding by an atmosphere or not any other kind of shielding. And as I mentioned before, we were using this germicidal radiation. And also another important part is we made experiments under sodium light. And this is to prevent one of the cells are able to use one of the mechanisms to repair DNA. There are two mechanisms, and one is depending on light. So we wanted to prevent this, so to put the cells in a weaker condition. This is just to show you how we count the survival after the irradiation. We use these plates and we count these dots, which are colonies. These are called CFU or UFC, depending. Colony forming units in English, CFU. So we consider the number of colonies of non-exposed cells, which is the control, and the number of colonies after irradiation. And this number is giving the survival fraction. Usually, this is what I was mentioning some slides before. It's the logarithm of this number as a function of the fluency. And this is a survival fraction. So with this, these are the results of the approximation paper as an example. And here you have the survival curves. This is for the flare case, and this is for the super flare. And this is for haloferrax, and this is haloferrax and salmon. What you see here is that these curves are having two slopes. This is meaning that for the lower fluencies, the survival is the decay in the survival is very fast, very quick. And then you have this tail, and you see the same shapes independently if you have a flare or super flare case, and independently of the microorganism. So it seems to be something general, but the most important thing is what is this tail, and this tail is indicating that there is a subpopulations of cells which are more tolerant than these ones, that the ones which are represented here, this part of the curve, and these cells are tolerant to UV. They are able to tolerate higher fluencies. Here in this is the time, but the fluence. And in this case, this was lower because also you have to consider here that there is a limit of detection of the technique, and then these microorganisms were reaching this limit like quicker or at the lower level of fluence, and that's why we don't see cells anymore. But the fact of not seeing survivors or not able to count them is not meaning that they are not existing, just that there is a limitation. And the limitation is given by the technique. Well, the conclusions of this is that This subpopulation of microorganisms, as I was mentioning before, is able to tolerate these high UVC fluencies, which are related to flare or superflares. And the presence of the subpopulations is also independent of the tolerance of microorganisms, because it's disregarding which microorganism you have this state. And here this plot is showing the effect of the photo repair with some experiments with sodium light and some others, added lights. My purpose that the microorganisms are able to repair, and at least with this microorganism we didn't find big differences. So it would be the same, more or less, if the microorganisms are able to repair or not, you would see these states anyway. And we obtained similar results for TRAPPIST-1 paper, or the Iococcus radiodurans, and the paper which is submitted now to notices. And here, well, comparing the results then, the study before, they said no survival for a superflare case. But here we said, in base of our experiments, that we had survival even to 40 kilojoules per square meter, in comparison with this value, which was shown as a limit. This is 72 times higher. And also for less radiotolerant microorganisms than the Iococcus radiodurans. And with the other paper, the TRAPPIST-1, well, as I was mentioning before, these were the results, survival in some cases and some other not. But our experiments were showing survival for every of the cases that we analyzed. So this is meaning that the previous theoretical studies were underestimating the chances of microbial life to cope with these radiation fluencies. And why is this? Well, now this is having this about these values they were calculating. The problem is when you take these values with experiments which were done at low fluencies, you would only see this part of the curve and you will not see this one. So you would take your parameters here, but these parameters are not going to represent what is happening at high fluencies. And this is the main issue of these theoretical approximations. And yes, in some cases we have 3 phases in the curves or 2 phases, but yes, in both cases it applies the same case. I mean, for super flares or flares. So the thing we are learning from this is that we shouldn't extrapolate from low fluencies or low fluence rates to high fluencies. Yes, now is the second part of the talk. I wanted to mention a little bit about this project we are having with saline and hypersaline environments in the solar system. We are having these very beautiful environments on Earth, these hypersaline environments, for instance, the sea. And in the past, it was thought that these places are not having the possibility to contain life. They are looking quite lifeless. You don't see animals or plants, and then you may think there is nothing there. But then, well, before that, we have these other kinds of, we can have also salt flats like this. They are looking like very lifeless as well, or these salt caves. And what I wanted to say then is that this was the idea until 1913, where the first microorganisms, halophilic microorganisms, were isolated from these hypersaline places. And these were isolated from the salt mine in Poland. And from then on, many studies tried to isolate microorganisms from this kind of environments. We are having these microorganisms which are called halophiles, which are living in these environments, and we are having different levels of tolerance. The most tolerant are the extreme halophiles, typically they are halophilic archaea. And well, this is a place in the Atacama. You have this very nice hypersaline habitats. You can have these salt flats, but also you can have these hypersaline lakes. And what you see here is not sand, they are salts. And if you watch carefully, you see these colors, and these colors are connected with the presence of microorganisms. For instance, the green cyanobacteria, halophilic cyanobacteria, or halophilic archaea. If the concentration of microorganisms is really high, you even can see these beautiful colors in the water, and this is connected with the presence of these microorganisms which are containing pigments. And well, this is just to show different kinds of environments. We are having this microorganism in our lab, which was isolated from the sea. Also, this one, which was isolated from this lake in Kenya. You also have different pH conditions. This is neutral and this is alkaline. But you can also have acidic environments. And also another important feature of these hypersaline environments is that they are considered Mars analog. We have here our colleague, collaborator, Cecilia. She is studying these kinds of environments. For instance, they were developing these environments where they were finding, for instance, native sulfur. This is a very recent research on the surface of Mars. Also, they were finding this sulfur on the surface. And also you can study these environments, of course, from the microbiological point of view. And then one could make some comparisons or extrapolations about what could happen in other planets. And also these environments are interesting, and these microorganisms are interesting because of something I was mentioning at the beginning of the talk. We are having these microorganisms there, which are having mechanisms for tolerating quite well the radiation, in particular UV, photoprotection mechanisms, special mechanisms to cope with DNA damage, and also mechanisms for DNA repair. And there is an interesting mineral found in these hypersaline environments, which are called highlights. These are sodium chloride minerals. You have these very big minerals. This is, for instance, in the Natural History Museum in Vienna. And this is called also blue halite. This is halite which was subjected naturally to gamma radiation. You have this blue halite. And in general, you have this kind of crystals. They are square shapes, different sizes, microcrystals or big crystals. And how these crystals are formed? Well, let's think in the case we are having a body of a lake, hypersaline lake. And these hypersaline lakes, depending on the season, sometimes are evaporating, and we have desiccation. And then they are having periods of fluid again, and what is happening is, like, if we have microorganisms living in these hypersaline lakes, these crystals are formed, but the crystals are entrapping these life forms inside. And this is what you see in these pictures here. These are from different papers. Here you see these kind of bubbles inside the crystals, and these are called fluid inclusions. The fluid inclusions could have liquid or gas or a mixture of both. And inside these fluid inclusions, you find these microbial life forms. This is very interesting because we are having very ancient highlights. And some studies isolated microorganisms from these very ancient salt crystals. And this is making us think in the capability of these such crystals to preserve life for extended period of time. Also, we are having extraterrestrial highlight. And one example is, for instance, the highlight in the Nakla meteorite. This is a Martian meteorite, where you could see this highlight, the presence of highlight inside the meteorite. But also now we are having, with the development of missions to different planetary bodies of our solar system, we are able to explore. And there are also salt flats on the surface of Mars proposed. That's through different missions. They were able to recognize these hydrosaline places. But also now with increasing number of missions to other planetary bodies, they are able also to see that these environments are also extended to other moons of the solar system as well. It is even more relevant. And as part of our program, what we are doing is again these laboratory simulations, and we are trying to reproduce these highlight crystals in the lab and trying to reproduce which processes could happen in the extraterrestrial environments with these salts, and how these salts could protect life, for instance. This is some images from some experiments with the microcrystals we created in the lab. And here you can see microorganisms inside. This is they were stained here, and this is under confocal microscope. And we are able to see the microorganisms inside the crystals. Also, previous studies from other authors proposed that the highlights, for instance, can confer protection against UV for halophilic microorganisms. And we took this idea to extend these studies, also including some astrophysical aspects. And this is one of the studies we published in the past year, where we combined two different projects, and we studied the protective effects of these minerals in to considering vacuum and vacuum ultraviolet radiation. And we considered superflare, in particular a case scenario which would involve the early solar system. And this is because we were studying here like planetary surfaces which are having atmosphere, like airless bodies or meteorites. And this is a study we published in Astrobiology. This is a nice cover of the journal, which is showing our study. And well, we consider here for calculating the UV fluxes of the young sun, we work with this capacity. This is something the colleagues here from Graz did. And we studied this at, we made calculations of the orbit of Earth, Mars, and Venus. And we were working with this vacuum UV range and vacuum in these levels. And here we compared these minerals, these salt crystals made of sodium chloride versus crystals which are made with other composition, like with salt mixtures. And also comparing these with cells which were unprotected. So this is meaning like they were not inside the salt crystals. And we did this for the Inococcus granularans and Alliferans vulcani. And in this case, the laboratory simulations were implying the synchrotron accelerator. This is a facility in Brazil, unfortunately not available anymore. But we were using this facility to make our experiments to simulate the vacuum UV, these vacuum UV levels, and also vacuum. Some of the results we were having, so like the summary of the results, we saw that when microorganisms are inside highlight, they are having a higher survival to vacuum in comparison with the organisms which were trapped inside these crystals, this oil composition. And also we saw the same for vacuum UV. So this is a plot which is showing the survivors as a function of influence. This is for the Enococcus radiolurans. This is like the cells, the unprotected cells. These are the cells in highlight, and these are the cells in the hard mixture crystals. And we see that the survival is higher when you have these highlight crystals. And this is for the other the halophilic microorganisms. Interestingly, for us, this was a side result. We were surprised that the Enococcus radiolurans is a non-halophilic microorganism, however, was tolerating very well the entrapment, and also they were the effect you see is even stronger for the Enococcus radiolurans than for the halophilic bacteria. So the main conclusion of the paper is that these subcrystals seem to confer a better protection than crystals made of a different composition. And even when this mixture of salts is having an optimum condition for the haloarchaea, because this was trying to mimic the mixture of salts where these microorganisms are living naturally in the terrestrial environments. And another part of this project is involving these laboratory simulations as well. And we started this in 2014 to make these simulations with salts. We were starting to make these experiments in the Astro Lab in Brazil, in connection with the same colleagues I was working with at this synchrotron facility. And we were publishing some results already in 2016, but we are continuing now, as Gunther mentioned, here in the Astro Lab. And this year we were continuing with some simulation experiments using the facilities here. And what is more interesting is that we could make some kind of inter-laboratory comparison, because of course, there are a lot of limitations of these experiments. It's not sometimes so easy to simulate these environments and also to study the samples afterwards. So this was very important for us because we could compare the facilities, and we could watch what is more convenient for this kind of experiments, also to improve these kind of chambers in the future. Some other developments we are currently starting to develop now is to work with other kinds of radiation. This is a facility which is existing in Argentina of our National Commission of Atomic Energy. We are having this tandem accelerator, it's called TANGAR. And this is the view of the accelerator from outside. This is from inside. We are going to start to make experiments with protons and high ions. And also inside this condition, we are having also facilities which will allow us to make experiments with neutrons and gamma rays. So we are trying to expand our studies now to other wavelengths as well. So, well, to finish, some summary, and maybe we can have a discussion afterwards. After my point of view, also biological experiments in an astrophysical context are very complex and may have a lot of limitations. I believe they could represent a better approximation of what will happen in a real extraterrestrial environment. We are also thinking that these biological studies we are doing involving experiments can help even to improve these theoretical studies. also to provide them with new parameters and constraints. And these laboratory simulations offer like a reliable approximation in comparison to experiments, for instance, for the study of these scenarios. And it would be very important as well that to work in the development of facilities and instruments for these habitability studies, and this is part of what we are doing. And also, I consider that interdisciplinary approaches and teams are having advantages with respect to intradisciplinary studies. And it is more important for these habitability studies. Of course, one could discuss advantages and disadvantages of different approaches. Sometimes the theoretical approaches are even useful because one could not reproduce every condition in the lab. So at the end, I consider that also a combination of different approaches is what is giving us at the end a better approximation for to study these extraterrestrial environments and these habitability stars. We can discuss this afterwards. Well, thank you, and I'm open to questions. And thank you to all my collaborators.